Welcome everyone to today's webinar. I am Brian Zimmerman with Becker's Hospital Review. We will begin today's webinar with a presentation and we'll have time at the end of the hour for a question and answer session. You can submit any questions you have throughout the webinar by typing them into your control panel in the space labeled Enter a Question for Staff and clicking Send. We are looking forward to hearing your questions. Additionally, in about a week following the webinar, we will be sending you a copy of the presentation to the email you used to register. At this time, it is now my pleasure to start today's webinar by introducing our first presenter, Nabil Hafez. Nabil brings extensive experience in product management, genomics, diagnostics, and informatics to Sunquest. Prior to Sunquest, he was Product Director, Neurology at Quest Diagnostics, Director, Clinical Strategy at GenomeQuest, Founder, SecWise, and NGC Consultancy. Nabil began his career at Broad Institute. Nabil holds an MBA from Duke, an MS in Bioinformatics from Brandeis, and a BS in Biology from Binghamton. Nabil, I'll now turn the floor over to you.
Thank you, Brian. I am very thrilled to introduce our expert panelists joining me here today that we are honored to have. Dr. Anthony Cerecci is Assistant Professor of Pathology and Cell Biology at Columbia University, as well as Assistant Medical Director of the Laboratory of Personalized Genomic Medicine. He also holds the role of Assistant Director of Biochemical Genetics. Dr. Cerecci's clinical practice is centered around molecular pathology with a particular emphasis in cancer genomics. He is an active member of the Association for Molecular Pathology, where he lends his knowledge and experience in payer and government policies as the group's Vice Chair of Economic Affairs. Dr. Cerecci holds an MD from Johns Hopkins and has completed his clinical pathology residency at Columbia, where he also earned a master's degree in biostatistics. I'm also so thrilled to be introducing Dr. Matthew Lebo, who's head of bioinformatics at Partners Personalized Medicine, a division of the Harvard-affiliated Partners Healthcare. Dr. Lebo began his work at Partners Personalized Medicine as Associate Director for the Laboratory of Molecular Medicine, working to enable the clinical adoption of personalized genetic medicine. Dr. Lebo holds a PhD in computational biology and bioinformatics from USC, completed his fellowship in molecular genetics at Harvard Medical School, and is certified by both the American Board of Medical Genetics as well as the American College of Medical Genetics. I'm going to give a brief overview of precision medicine pertaining to our topic here, informatics to drive precision medicine, and then we're going to turn this over to the panel discussion as we proceed through here. So there's lots of headlines around precision medicine and the growth of precision medicine that many of you have probably read, the explosive growth, the promise of precision medicine. There's a lot of hype, a lot of headlines, but there's also a lot of reality around what's happening today with the FDA approving many medications this past year alone related to precision medicine treatments and precision medicine diagnostics. It is estimated that 1/3 of diagnostics will be molecular or genetics based over the coming years. If you ask people what precision medicine is, you'll likely get a bunch of words related to genetics, patients, cancer, and treatments. For the purposes of today's discussion, we're going to say that precision medicine is using molecular medicine and genetics to diagnose, classify, and treat patients. Next slide, please. There's been a rapid rise in the nature of molecular and genetic testing, especially at laboratories and physicians that have been ordering this type of testing. 5 years ago, there were around 785 conditions that were being tested with genetics. Now over 9,700 conditions have some sort of genetic test related to them. In 2016, 49,000 clinical genetic tests were offered by the nation's laboratories. Today, the number is well over 60,000 tests are available to order from laboratories. So at the same time that the number of testing is growing, the growth of data, the literature, the explosion of data generated externally and internally is also growing. Over the past 14 years, the number of genetic-related articles has increased 8-fold, And what's astonishing is the amount of data that a genetics or a sequencing instrument is producing per day has exponentially increased over 60,000-fold in the past 12 years. No other technology has advanced at such a rapid pace. Next slide, please. But there's real challenges in enabling precision medicine as part of mainstream part of healthcare. There's a lot of challenges related to laboratories that are performing clinical testing and genetics testing. So if you were to compare clinical testing and routine CBC testing, that really hasn't changed in the past 40 or so years, where there's 2 to 3 steps. You acquire the sample, do some minimal sample preparation, and put it on the instrument to get the result. Compared to molecular and genetic testing, it's a lot more complex for the laboratory with over 10 to 20 steps. And the technology itself is constantly changing. Many times it's outdated in months with the constant change. There's significant reimbursement uncertainty around some of this testing for in some areas of application, such as cancer genetics. It's sometimes better than other areas, but there still remains challenges related to reimbursement. And then when you look at the data analysis challenge, if you were to sequence a human genome, you're likely to obtain 3 to 5 million variants per genome. It's estimated that 83% of the variants that are obtained from this are what's classified as variants of unknown significance, or where there's not a lot of information known about them. At the end of the day, the clinician, the pathologist, needs to deliver an actionable report for the treating clinician to be able to diagnose and treat their patients. This takes a lot of effort, and it's estimated that 55% of labs' next-generation sequencing genetic costs will be allocated to biological interpretation by 2020. It takes on average 1 hour to assay each one of these variants and determine each one of these unknown variants and determine which ones are applicable for that patient and for that physician treating the patient. So how can laboratories keep up with all the external and internal data and some of the challenges we discussed? And that's the topic of today's conversation. So with that, we're going to get into the panelist conversation here. Next slide, please. So I'm going to ask Dr. Surechi can you please give us a little bit of information overview about your laboratory and institution, please?
Sure. So first of all, thank you for the introduction. Again, my name is Anthony Surechi. I'm at Columbia University Medical Center, and I'm assistant director of the Laboratory of Personalized Genomic Medicine. So our laboratory sits squarely in the university and provides testing for NYPH Columbia patients only. So Cornell, which is our sister institution to the south, has its own molecular laboratory and offers testing to NYP Cornell patients. We are a full-service molecular laboratory offering genetic diagnostic assays in a CLIA-approved, New York State-approved laboratory across multiple service lines, including germline or constitutional testing, prenatal testing, cancer genomics testing, and some microbiology testing as well, molecular microbiology testing. We run approximately 50,000 samples a year. The majority of those samples are, in fact, microbiology. But we've also run, by now, probably about 500 whole exome on cancer patients, over 1,000 constitutional whole exomes, and we also offer a variety of panels for cancer genetic testing, including a 500 gene panel, which is sort of similar to that offered by Foundation Medicine. Our physicians are generally fairly high functioning physicians in terms of genomics. So our oncologists, we have a very active phase 1 program here in oncology, and on the constitutional and prenatal side, we have a very strong genetics and genetic counseling group here who receive our results.
Great. And how many people would you say are affiliated with your genetics program at Columbia?
The laboratory has a staff of 7 full-time faculty, with some part-time faculty distributed among some of the researchers with subject matter expertise and our surgical pathologists with tumor type expertise. And then the laboratory itself also has a staff of about 30 individuals, with 4 of those individuals dedicated full-time to bioinformatics, which is actually a fairly lean bioinformatics team given our volume and complexity of our laboratory. And then as a whole, the phase one group in both, there's a phase 1 group in both adults and pediatrics, and there's also a full department of genetics that sits in pediatrics, I think with 4 full-time attendings and genetic counselors and whatnot.
Great. Thank you, Dr. Strachey. Dr. Liebel, can you tell us a little bit about your institution and laboratory as well, please?
Sure, yeah. So I work at Partners Personalized Medicine, which is a personalized medicine center that is a sort of joint between Partners, the Brigham and Women's Hospital, and Mass General Hospital. And the center has sort of 3 main cores. It has the clinical lab, which is a lab for molecular medicine, which I'm also now director of. It has the translational genomics core, where we do sequencing for both our internal samples and other people within the institutions, and a biobank that's across the partners institutions as well. And then I direct the bioinformatics team, which supports all 3 of those endeavors and a few of our other grant-related activities. Our lab is mostly a reference lab. It's been in operation since early 2000s, so about 15 years or so now. Traditionally started with panel-based testing for cardiomyopathy, hearing loss, Noonan syndrome, and a few other disorders like that. More recently have been doing exome and genome sequencing, and have been doing a lot of sort of healthy individual screening. So we've done a lot, all the sort of interpretation-based work for part of the eMERGE consortium. They have a new sequencing project that's going on now. For the Geisinger MyCode project, we've done those interpretations, and a few other large-scale projects similar to that. And yeah, so and then the bioinformatics team that I oversee is about 9 people, I think 9 or 10 people. We also have a pretty large IT staff. There are, I think, 6 PhD geneticists who have other roles as well, 3 genetic counselors, and then another plethora of sort of lab staff and or with clinical interpretive staff.
Great. Thank you, Dr. Liebel. Can you tell me, can we go to the next slide, please? Why did you decide it was important for your institution to create a precision medicine or genetic testing program? And how did you get to making it a reality over the past 10 or so years?
Yeah, so I mean, initially, the center has undergone a few name changes and a few sort of change in focus, but it was initially seen as around 15 years ago that there was a partnership between Partners Healthcare and Harvard Med School to sort of develop a joint personalized medicine center. And that actually led to some of the early initial genetic testing. So we were the first lab to launch cardiomyopathy because we had a very deep interest in cardiomyopathy at the Brigham and Women's Hospital. We're also the first lab to launch EGFR testing for lung cancer because we had a close collaboration with the MGH scientists that developed it. So it was built to be sort of a leading edge lab in terms of both the informatics that we were doing and some of the laboratory assays. And more recently, our focus has shifted to even being more focused heavily on some of the informatic aspects and sort of how we can manage to get some of this stuff back into the EHR system.
Great. And I think both of you talked about the size of your staff and the number of clinical geneticists and genetic counselors that you have on staff. Can you speak to a little bit about institutions who are now trying to start a precision medicine program? Is all of that required to get into genetic testing, basic genetic testing today? Maybe Dr. Strachey, can you comment on that a little bit?
Yeah, so we started out in molecular pathology around 1999. Some of what Matt was just describing in sort of the age of PCR and real-time PCR, just when molecular diagnostics was entering the mainstream diagnostics. When we moved into genomics testing, so next generation sequencing based testing was also very early on, around 2011, 2012. And at that point, there weren't a lot of options other than to build the infrastructure yourself. So to purchase the sequencers, get the technical know-how to process them and understand sort of the vagaries of the technology, to build a bioinformatics team that would work with physicians to develop a bioinformatics pipeline for variant interpretation, or for data processing and variant interpretation, and to deal with how the data gets from the pathologist to the medical record. So we had to build all that from scratch, in addition to building the sort of the knowledge base of how to interpret variants. Over time, and since we started, as I said, some 5-6 years ago, there are more and more commercial opportunities for that, so that a laboratory that's just starting today has many, many more options on the buy side rather than the build side. So I would say there are, number one, there are molecular limb systems that help you get specimens into the laboratory accession in a more traditional workflow, that we're comfortable with in the clinical laboratory, that just didn't exist when we first started doing this type of testing. There are also commercially available pipelines that would take the data, sometimes even directly from your sequencer, and then do the alignment, the variant calling, and may even provide some interpretive comments for you. It depends on what the system is. And finally, there are report generation and knowledge bases that are also purchasable. And so some of these functionalities overlap in one system and some do not. And the final purchase possibility is also systems that take the genomic data and then marry it to the EMR, making it far more powerful in terms of identifying new phenotype, genotype correlations and recruiting patients to clinical trials. So all of that now is purchasable. The real question becomes, what is the cost of building it yourself versus the cost of buying something that's, out of the box and that might require a little bit of finagling, but is already sort of ready to use. And that really depends on the types of questions your laboratory is asking. If you're trying to push the envelope and do new cutting edge type work, then maybe the systems that are purchased are not for you. But if what you're trying to do is the diagnostic work, then I would argue that there's a pretty strong argument to be made for the purchase option.
And I'd also add, sort of on the underlying hardware, it's very similar. When we first launched NGS testing too, we had to do everything in-house. So we have our own HPC system, and we use the hospital staff to sort of support that. But now there's so many more cloud vendors out there that you don't necessarily need to do that. And even some of the pipeline activity is coupled much more directly to the cloud. So if you were to sort of do the buy decision, you would actually have the ability to work in the cloud too, and not have to worry about storage. But it sort of, again, depends upon the institution and how much they care about having the data on premises or the expertise on premises, whether or not you sort of, from the computational perspective, do that sort of in-house or go up to the cloud.
So Matt, that's actually an interesting point. We faced a lot of pushback from our institution from the data privacy and safety people who were very against using cloud-based solutions, and they're just now starting to come to terms with the fact that such high volume data may actually not be possible to process locally on local servers, and may require cloud processing. I wonder what your experience was like with your data security and safety people.
Yeah, it's the same thing. Initially, when they were doing this, they had not for any other sort of applications either actually gone to the cloud, but they've since have some applications out on the cloud, but still at a low level. And so there is a little bit more appetite. I think from us right now, it's sort of figuring out how to scale this. So we have a large project coming through where there's going to be like hundreds of genomes that we're trying to process at once, and doing that on premises would just take a huge purchase from an infrastructure standpoint. And so it actually is getting to be about the time where we have to think about other ways for us to actually do some of this within a cloud-based framework, even if it's just this upstream analysis and we still pull the data back and maintain it and store it here.
And how do you both deal with the challenge of, you just both touched on it, with EMR connectivity and returning results to those clinicians that consume the data that your laboratory is generating? Dr. Lebo?
Yeah, so we have so right now for our institution we use GeneInsight, which was a tool that actually was developed in-house locally and has since been purchased by Sunquest. And while it was still built here locally, we developed an application that sits within our EHR system. So we have Epic, and we built a sort of an interface within the system that can receive reports from our system. If we sort of change the interpretation on the variants, the reports, the alerts get sent out, and there's information associated to the reports that the information on the variants has changed and what the updated information is. So if we were to change a variant from uncertain significance to pathogenic, and an alert would get sent out to the clinician so that they would know, and then it would also be reflected within the genetic screen of the EMR.
Great. And Dr. Strachi, how do you cope with that at your institution?
We report all of our results through Cerner CoPath, much like our anatomic pathology laboratory does. So our reports follow the same structure as a surgical pathology report, and they get pushed into our multiple EMRs downstream of the laboratory in a text file format. As you can imagine, that's not ideal for clinical trial recruitment. Say you have a trial coming up looking at PIK3CA variants, and you want to know, approximately for a feasibility study, how many patients this year were diagnosed with a PIK3CA variant. That's not doable from the EMR. That has to be done through our pathology database. So it's not an ideal system, but it's a system that we've developed, as an offshoot of our standing limb system, which is Cerner CoPath. We are migrating to Epic. NYPH, both Columbia and Cornell are migrating to Epic in the coming year, I believe. And so there will be a lot of effort put into thinking through how our results, which are very structured actually, it's always templated, how those results might be parsed out in Epic into a searchable format and then stored in a clinical data warehouse for clinicians to query on a research basis.
Great. And Dr. Lebo, can you tell me a little bit about when you first started offering genetic testing, how that was received by your clinicians, and now when you offer it, it has kind of the learning, the education required to understand this testing evolved, or have they become more savvy or more educated, or is the kind of what you're reporting become more actionable and informative to help them with the adoption of this type of testing?
Yeah, so that's a good question. So, I mean, initially when we started doing this, it was much smaller gene panels. Like they were Sanger-based. There was maybe 5 at the most, and they were ordered primarily by pretty savvy clinicians. As the testing field grew, not only did the sort of scope of the assay grow, but also the amount of stuff. So the amount of stuff that we looked at grew, the complexity grew, but also sort of I think the general knowledge grew, and more people outside of the sort of even specialists with genetics understanding would be ordering the test. I think for us the biggest difference came in when we launched NGS testing in 2011 and went from like 10 genes to 50 or so genes, and the number of variants of uncertain significance, and clinicians now getting those back, but also in our laboratory, how we're going to figure out how to report them and how to manage them, because the scope of them is just so much greater. Since then, we definitely have gotten a better handle on it. We have a good database behind everything that we've classified, so we have a good understanding of what we've seen before and what we haven't seen before. So I'd say that I think the testing has sort of plateaued. I think people understand that for an exome in a genome, you do not return every sort of variant of uncertain significance, because that would be tens of thousands or millions of variants. So you really, the type of things that get returned are much different. So we used to even include initially benign variants on our reports. Up until recently, we included likely benign variants on our reports. And we're definitely moving away from that model, where even in our exome and genome, it's primarily just likely pathogenic and pathogenic variants that we're reporting. So as the scope of the assays has increased, we've had to sort of figure out ways to deliver back the most relevant data in each sort of instance. And so we have definitely changed along with the times and are doing a much more focused reporting scheme, even though a lot of that other data is sort of still kept in the background.
Great. Any comment related to returning actionable and information to your clinicians, Dr. Sureshi?
Yeah, I'll add that from the oncology perspective, it's just somewhat different. So that the paradigm that Matt described is exactly right. That when we were doing EGFR and KRAS testing alone, or BRAF testing, it was pretty easy for clinicians who were actually quite specialized in their organ areas to understand the results. Either the variant is there or it's not, or it's actionable or it's not. As we expanded to 500 gene panels and exome and transcriptome analysis for some of our pediatric tumors, the requirements for discussion really became much more palpable, so that even the expert clinicians weren't sure what a lot of the variants we were reporting meant. We've always taken the approach of tiering our variants in sort of conjunction with the AMP reporting guidelines, so tier 1 through tier 4 variants. Even with that, we've found that we get a lot of calls and questions from clinicians. So what we've done now is we've instituted molecular tumor boards, both for adults and pediatrics, so that a clinician can ask for the case to be discussed if there are variants that are of interest. We even have a germline tumor predisposition tumor board that, to which a patient can be referred. So there are opportunities for the molecular pathologist, the surgical pathologist, the treating oncologist, the surgeon, so on and so forth, to get into a room and really discuss the case. And that's almost required when you're doing larger panel testing, I think, in cancer, particularly at a site like Columbia, where there are so many clinical trials, it's hard to keep track of what's available, what's recruiting, and whatnot. So we found that to be particularly helpful.
Great, wonderful, thank you. Can we go to the next slide, please? So we discussed some of the challenges that you faced related to data. Dr. Lebo, what advice would you give someone or an institution who's starting their own precision medicine program today?
Yeah, so I think the biggest thing is to really figure out the scope of what they're trying to do, and figure out sort of the reasons behind it. So Anthony and I both work at academic medical centers, and so not everything, but a lot of what drives some of the things we do is sort of the data behind some of the tests. And so actually building the repositories, building the database, collecting the samples, that's just as important as sort of some of the results that we're getting. And so to be able to want to build that infrastructure, you actually want to make sure you want to do that from the outset. And so that also gets into sort of the build versus buy decision too, understanding what you need to bring in-house in order to support a precision medicine program. Whether you need all the expertise, where you can outsource some of them, what aspects of the testing or the information you want to keep in-house, and what sort of experts you want to bring on board. I think it really depends upon what the general mission of the program would be. Great.
And same question to you, Dr. Sureshi. What advice would you give to someone starting a precision medicine program today?
So 100% echo what Matt said. I would also say that the economics of the discussion really needs to be an economic discussion that's pretty frank needs to be had. So questions around what are the actual true costs of a precision medicine program what are the true costs associated with building such a program? And I think, we focus so much on the diagnostics portion, but there's an infrastructure that exists in the pre-analytical, post-analytical side of things that don't include the laboratory. So genetic counseling, return of results, appropriate filing into the EMR, appropriate follow-up. There's just a lot of moving pieces that need to be discussed that are even outside the laboratory. That's not to say that the laboratory investment isn't critical and quite substantial. So if you're not already offering a next-generation sequencing platform, bringing it on for one or two disease states doesn't usually make a lot of sense, because then your fixed costs are really only distributed across those one or two service lines. What you really want to think about is a more holistic approach where you distribute, where you're able to distribute those fixed costs, which can be quite substantial, across multiple tests or multiple diseases, so on and so forth. And the other thing I would think of is on the flip side of the economic discussion, reimbursement. So how do you recover those costs? So having a real look at your payer mix, looking at the CPT codes you would bill for the variety of diagnostic services that you're considering bringing on, and then looking at the coverage policies for each and every one of those payers, and your contracts with those payers, because I will say they vary broadly, and the coverage determinations by Medicare don't always get aren't always reflected in private payer policies as well. So there may be opportunity both at the contracting level or at the advocacy level to change that, but you have to know sort of before you dive head in and spend the money.
Great. And Dr. Liebel, how is your institution addressing some of these challenges related to the economics and the reimbursement around genetic testing?
Yeah, so that's it's definitely a challenge. I mean, so you can probably tell from a lot of what I said, we primarily focus on the constitutional areas, and it is actually much harder in the constitutional space to get reimbursement right now. There's some CPT coding associated with some of the tests, but most of the sort of germline-based tests don't actually have specific CPT coding, and so there's definitely challenges with reimbursement. One of the things we've tried to do, especially for some of the more expensive tests like our exome and genome, is institute a pre-authorization policy where we work with the clinician and with the patient to try to understand, or to write the pre-auth letters we have to try to go that route to make sure that there actually will be the reimbursement before we sort of go down the path of figuring out how to pay for the expensive test.
Great, thank you. Are there other challenges that you've both faced as you started taking on precision medicine and scaling it and trying to grow it at your laboratory that maybe we haven't touched upon here so far? I know we've talked about the EHR connectivity piece, we talked about reimbursement, and we touched a little bit about the data challenges. Dr. Sureshi, what else did you encounter when you tried to do this type of testing at your institution?
Yeah, so I will answer that question in a second. I want to say one point about the pre-authorization. So, we do the same thing. It is new for pathology to be involved in something like that. We don't normally have that infrastructure in place, so we've had to partner with our faculty practice organization to help us do that. It delays testing. It certainly is not a physician satisfier, but it is a reality, and many insurers, including UHC, require pre-authorization for all genetic testing. I think that's going to be a pattern of behavior. So figuring that out and figuring out a system that works for you and works for your institution is going to be key. That's all I'll say about that. So the other challenge that we found, and this is not unique to precision medicine, but it is sort of unique because of the scale and speed at which it happens, is the requests for novel diagnostics and for new test development are sometimes out of control. So, ASCO will come around, and we have a group of 20 oncologists who go to ASCO, and then they come back with, requests for, their diagnostic du jour. And while I appreciate that science has to move forward and diagnostics has to sort of follow quickly, there's oftentimes a little bit of a disconnect between what's actually clinically useful, what's going to get paid for by payers, and what an oncologist will order and what an oncologist thinks that is right for the patient. So we've addressed that at Columbia by having essentially a new test committee that's made up of, it's not just in precision medicine, it's across the diagnostic laboratories, made up of clinicians, pathologists, laboratory administrators, specialists in coding and reimbursement. And so really having a full, thorough discussion about what the strategic goals of the laboratory are, and if new diagnostics that are requested fit within that strategy, and whether they're appropriate for bringing on. Because I'm sure, Massachusetts is a heavily regulated state as well. New York State is particularly onerous because of our Department of Health requiring submission of dossiers for every assay that we bring on. So it's a time-intensive, documentation-intensive process, and so we want to make sure we're expending resources appropriately.
Yeah, I agree. I mean, one of the things that we're doing now is to mitigate that, again, since we are much more concentrated on the germline, is actually using sort of a single backbone approach. So even for all patients, we'll run the same assay, regardless sort of what their indication might be, and then we can bioinformatically limit what we're looking at. And we can also then bioinformatically expand the test if we need to, and then the validation is not on the wet lab side anymore, it's just much more on the bioinformatics side, which is definitely easier. And Nabil, one last thing I'll bring up that was definitely difficult for us, and still is difficult for us, is and you mentioned this before, the time it takes to do the interpretation and the cost it takes to do the interpretation. So the interpretive staff is very expensive staff usually. Oftentimes it is geneticists, genetic counselors, other sort of PhD level people or MD level people, and it is right now fairly time consuming. There's not great software out there that can fit a lot of our needs, and so the time it takes to actually do the interpretation is quite intensive, and so it's something that sort of shouldn't be forgotten. And if someone's looking to build a program and figure out how to do this, trying to figure out the best way to streamline that process is actually really important. And it also is something that the field is figuring out ways to do it, but it still is very manual. And so trying to make sure that you have that in place and figure out how to do that is, like I said, very, very important for being able to do this successfully.
Great, thank you. And that actually flows right into our next question, if we can change the slide, please. So with all of the information out there to help you do that interpretation, and we talked about the explosion in external data. I think there's at last count, over 1,500 molecular or genetic databases that hold information related to disease conditions out there. How do you provide the most up-to-date and accurate interpretations of your genetic testing and ensure, you're not missing anything? Dr. Stretchy, can you comment on that?
Sure. I will just add one more point to what Matt was saying about the professional interpretation. So, pathology slides take interpretation, right? They take time to interpret. What makes genetic testing different is that CMS has decided to value them specifically only on the clinical lab fee schedule, so only technical reimbursement, no professional component to reimbursement exists yet for genetic testing. So pathologists don't get RVUs for it. It's there is no professional component whatsoever. So there's a real misunderstanding, I think, on the part of the payers, or willful blindness to the fact that it actually sometimes can take hours to review and diagnose patients based on genomics assays. So it is an unfortunate hole in the process there. So you're asking how we stay up to date. The answer for us is a mix of bioinformatics, so having our team update on a regular scheduled basis the databases from which our pipelines draw, and those are both constitutional for filtering out common variants in the population, and then cancer for identifying hotspot and targetable mutations in the tumor, and also falls very heavily upon the pathologists or laboratory directors reviewing and signing up the cases. So, that if keeping an eye out for genes of interest in particular tumor types, genes that have been associated with clinical trials, whether here or in New York City or through the match, which we're a part of, staying up to date in the literature. We really depend very heavily on our professional interpretation to mitigate any holes that are present in our bioinformatics pipeline. That said, we update our bioinformatics pipelines, I think, quarterly and version them appropriately, so that we're always sure we're pretty close to as up to date as possible. On the constitutional side, we offer reevaluation of our exomes and panel tests if a new clinical phenotype emerges, or if a physician feels that the classification of the variant that was issued by the laboratory may have changed over time, and we'll offer that free of charge.
Great, thank you. Dr. Leibel, you touched on that. Do you have more information you'd like, or commentary you'd like to add?
Just that for us, I think a lot of the standardization and data sharing initiatives that are out there are critical to this too. I mean, you can not be an expert in every gene in the genome or every region of the genome, and so having ways to sort of be able to use the expertise that are available in the community is incredibly important. So we're very strong and pro a lot of the data sharing initiatives. We were some of the major and first submitters to ClinVar and involved in the ClinGen project. And so a lot of those making sure that the community, and it is much more open now than it was when I started, but making sure that people are willing and open to sharing their knowledge, because it's really just going to make the process not only easier, but much more accurate down the line.
Great. Dr. Sureshi, do you use this type of data sharing at your facility?
Well, yeah, a lot of the databases are populated by data sharing. So, Matt mentioned ClinVar. We're also a part on the cancer side of the AACR Genie Cancer Consortium. That's going to be a huge opportunity in terms of sharing variants across multiple institutions, and then those variants become eventually publicly available. But there are other data sharing initiatives that we avail ourselves of. So CBio Portal, which is offered by hosted by Memorial Sloan Kettering, that is essentially a repository for those data. Cosmic, which is another data sharing website or database, which is accessible publicly as well. So absolutely, it's almost at this point, it's almost a requirement to stay to keep even up to date of the literature, more up to date than literature. It's sort of what are your colleagues seeing, and how are they describing variants? I think it's super helpful.
Wonderful. Okay, I think we'll transition here to the next question. Dr. Lebo, can you provide an example where in your laboratory you've seen precision medicine having an impact on patient outcomes that really wouldn't have been possible prior to kind of the advent of genetic testing, or your institution offering genetic testing? Do you have any examples that would be relevant?
Yeah, I mean, so one of the, I mean, there's many of them. So two sort of quick ones. So we do a lot of cardiomyopathy testing, and there was a case where it was actually a sperm donor who sort of passed on this variant to multiple individuals. And so being able to work with the different families, I mean, it's all probably consented, but work with them to get that information back, because these people would not have had a family history of cardiomyopathy. They could have sort of used that as a key to potentially they'd be at risk. So it really was their genetics that was driving it, but it was only found because of the genetic testing. And then the other one that we see a lot is we do a lot of hearing loss testing. And so there are certain hearing loss phenotypes where they respond very, very well to cochlear implants. And it's incredibly important to get it to these kids early, because the earlier you can get these implants, obviously the more successful they are. But it depends upon their type of hearing loss, which is oftentimes driven by the genetics. And so being able to figure out which kids are appropriate for cochlear implants and being able to get that into them early, obviously completely changes their lives. So.
Wonderful, thank you. Dr. Sureshi, how about at your institution, examples where precision medicine has impacted patient outcomes?
Yeah, so I like that Matt has the constitutional side, and I get to talk about it's a nice balance of where precision medicine has impact. So I can give you two stories, one in the adult setting and one in the pediatric setting. So in the adult setting, we had a patient who I think was a bladder tumor that had been sequenced or run by PCR and found to be microsatellite unstable, MSI. And MSI is one of the indications for providing Keytruda therapy, this new immuno-oncology drug that's available through Merck. But what we ran, so that's more standard type therapy, or more standard type testing on just PCR and size fragmentation. But when we ran our extended panel on this patient, we identified not only the MSI, which was true, but a variant in beta-2 microglobulin. And the way immunotherapy works is through the patient's own immune system. So beta-2 microglobulin, being a very important protein in the immune system, being mutated is actually one of the main mechanisms of resistance to immunotherapy. So the patient was spared a needless trial on immunotherapy that would not have worked because of our identification of their resistance mutation. So that's an example of avoiding therapy. We had a kiddo who had a rare, very aggressive sarcoma, who was tested by more traditional FISH methods. The sarcoma generally has a fusion in a protein called NTRK3. The more traditional methods, which are FISH-based, to identify the fusion with NTRK3 were negative. We did our whole exome and transcriptome analysis because it was a recurrent or treatment-resistant tumor. And we identified an NTRK fusion with a novel partner, so another partner to the fusion that would not have been picked up by our more traditional assays, and the patient was able to get a very new targeted therapy at the time, which targets this particular activating NTRK fusion, and was appropriately treated, and the tumor regressed. So that's an example of getting the right therapy because of precision oncology testing.
Wonderful. So one example where you essentially had the evidence to not perform the therapy because it would not have been effective, and the other that really was tailored to the patient in terms of their treatment.
That's right.
Great, wonderful. So with, I think, 13 minutes left here, we're going to switch over and open it up for question and answer session.
Yes, thank you all for that fantastic discussion. We will now begin today's audience question and answer session. Please submit any questions you have by typing them into your control panel in the space labeled Enter a Question for Staff and clicking Send. We will try to get through as many questions as we have time for. Got some good questions rolling in, and the first one here is, how can emergency departments benefit from precision medicine?
So yeah, I can talk about some of the things that we do. So we actually have some collaborations with the blood typing group here, and so actually we're working on some efforts now to sort of tie in some of the genetic prediction for blood typing with the actual platelets and everything that we have in house. And so ideally, what you can imagine is if you have everything typed appropriately, because you can get much more deep typing with genetics. And you can have a quick assay sort of in the ER. There's a very quick way you can match up sort of blood that's available to the patient that's at hand to make sure that you don't have any reaction there. And also we're doing a lot of sort of pharmacogenomic work too. And so again, similar, if you have a patient that comes in and they're at a high risk of certain drugs, I process codeine inappropriately, so it makes me really ill. So I wouldn't be able to handle a high dose, and so being able to understand that when someone comes in is actually critically important too. So I think the next step for genetics and precision medicine specifically is to actually go and have these genetic knowledge warehouses that are tied to the patients on the back end of the EHR systems, and when the patient comes in, that information is already available. And there's sort of apps that come up that sort of detail to the clinician exactly what to do in that scenario with that patient.
That's pretty cool. I like your idea about the blood typing. You know, in particular, if the person who asked the question has a high sickle cell population, patients who are already alloimmunized to red cells from multiple transfusions, I could see a huge impact for that type of testing. More general screening is not yet cost effective, but generally for patients who have seen multiple transfusion, cancer patients, chronically transfused patients, that's a great idea and a great utilization of resources. We should talk more about that. Very exciting. Yeah. So on the cancer side, we would interact very rarely with emergency departments, except in the case of acute leukemias, right? So if a patient with TML RARA positive acute leukemia, that clearly changes therapies. Similarly for AMLs that are newly diagnosed with particularly targeted therapies, a quick turnaround time for assays would be incredibly are incredibly useful, even at the level of the emergency department.
Thank you. That's helpful to know. Our next question here is, when should a health system start its own precision med lab versus outsourcing to third parties like Foundation Med? Won't these companies see a greater volume and hence benefit from economies of scale?
So I'll tackle that question first. So the answer is what Matt described before. It depends, right? So if the idea is to build the expertise in-house for a variety of reasons, whether that be academic, commercial, marketing reasons, there are multiple reasons why you might want to build these capabilities in-house, then outsourcing it sort of defeats those purposes, at least in the long term. That said, places like Foundation Medicine have huge amounts of data and can process your samples today, as opposed to the years that it'll take to build a proficient abilities in-house. In terms of the economies of scale, it's actually really interesting when you think about, particularly Foundation, and I think they offer a great service. Their costs are kind of weird, because whereas a lab like ours takes our infrastructure for NGS and spreads it across multiple service lines, multiple tests, Foundation Medicine offers one service line, right? Their foundation testing, their tumor testing. They have various assays, but they're all spread across this platform. So when we talked again about sort of diluting the overhead, the capital investments and the fixed costs, it all goes towards that assay. Whereas in a laboratory like mine, we sort of distribute that across multiple service lines. And so where they may have economies of scale on variable costs, those fixed costs are really not distributed appropriately. So I wonder, and I don't know, I've never done the analysis, but I wonder if you compared the total costs, if they weren't actually quite similar.
Great, thanks for that answer. Got some more questions coming in here. In today's market, what is valued most? The sequencing aspect, the report configuration/assimilation aspect, the interpretation aspect, or the clinical therapeutic workflow aspect? Asking in terms of where the biggest need is and where consumers will be attempting to fill gaps/voids. Thanks.
Yeah, so it's a tough question to answer. I think they all are obviously important and valued. I mean, I think that the sequencing is becoming and probably will become a commodity. There's obviously been reports about much cheaper and cheaper sequencers as they come out, and I think eventually there will be good enough sequencing that will sort of be easy to scale up and not as critical. I think it's the knowledge and then the delivery of that knowledge to the clinicians where the most value is going to be found, and how you can sort of manage that and do that appropriately is sort of the next big frontier of personalized medicine and of molecular genetics. And really, how do you do that within the full healthcare system? I think is where we see ourselves trying to go in the next few years. And I think it's going to be a big challenge, but it really is where there's going to be a lot of value found.
Yeah, I'd add that at least for actually, I think in general that connection between the molecular profile and outcomes, that I would say particularly in cancer, is still sort of this holy grail. This sort of how do we predict who recurs in patients with generally good prognosis tumors. There is always a small subset of patients who recur. One of the keys may be in the profile, both genetic, epigenetic, proteomic, sort of the marrying of the different databases with clinical outcomes to create a huge big data set that is then commercializable. I think would be a huge opportunity for anyone who's interested in sort of a more I guess, entrepreneurial, with a more entrepreneurial bent. And I'll also add that from our clinicians, what they want to know is how does the variant impact the patient's treatment, where are the clinical trials, and what therapeutics are potentially available to this patient. And that, to us, and I think that's the biggest value from our tumor boards and the type of data that we give our clinicians.
Thank you for those answers. And we've got a ton of great questions here. This audience member would like to know, I have some experience with regards to using genomic medicine in sequencing human papillomavirus. Do you have any other examples of the success of genomics in treatment of infectious diseases?
Yeah, I mean, sequencing, doing HIV genotypes, resistance mutations in HIV and HCV have, in the past at least, helped guide therapies in molecular microbiology. I don't know, Matt, anything else that you could think of?
I mean, so we're not doing any of this stuff yet, but on some of the research projects, we've been looking at some of the microbiome stuff and trying to figure out how that interplays with the human genetics. But I think it still is early stages for a lot of that, especially the interactions with the human genome. But I think that is obviously a place where the field will go eventually. It's just not quite there yet. So.
Great. Thanks for clearing that up. And this audience member would like to know, do either Dr. Srecky's or Dr. Lebo's labs have the capability to identify infectious disease pathogens and antibiotic resistance genes by molecular methods? If so, what methods / technology are currently being used?
So in our lab, sorry, Matt, go ahead.
I was gonna say we're not doing that currently today. We really are focused a lot on the germline stuff. I mean, we do identify cases where people are at risk, but we are not sequencing individuals with like depressed immune systems that frequently, where we get a lot of this information. So. Yeah,
well, our laboratory doesn't do that testing. We have a molecular micro laboratory on the hospital side of things who do. The type of viral genotyping that I described. I'm not sure that they're doing molecular testing for resistance mutations quite yet. But if that person wants to reach out to me, my email's on the website. I could try to put you in touch with our molecular micro people.
Right. Thank you both for clearing that up. And it looks like we have time for one more question here today. This is to either panelist. How long did it take for your lab to get a return on your investment in personalized medicine?
I mean, we set ours up so long ago, it would be hard to know. I think it was, I mean, it was not easy at first. If you're setting up a lab from scratch, it takes time. It takes time to order the instruments. It takes time to get the CLIA license. It takes time to sort of build up the expertise. And so it was a little bit of time for us. It was such a long time ago. It'd be hard to sort of say, if we were to do this today, how long we think it would take.
Yeah, I think it's a similar answer. We started so long ago, it's hard to say. But I would add some words of wisdom, which is the sort of like in a grocery store, you want your sort of low margin, high volume things that'll just sort of fly off the shelf. So that for us is our HPV testing, our cystic fibrosis testing, the sort of things where you're going to do a lot of it. You're not going to make a lot of money per sample, but you're going to do a lot of them, and that they sort of act as your cash cow for the more cash intensive things like NGS. If I were to look specifically at the business unit that does NGS, I would say we still haven't seen a return on that investment. But just because reimbursements are so low, you can't depend on clinical revenue for that type of work quite yet, unless you're doing institutional billing. But as a whole, as an investment in genomic diagnostics or molecular diagnostics, with the appropriate test menu and appropriate differentiation between your different service lines, you can actually make it work out, and you can have a net neutral or slightly profitable business.
Thank you for those answers. And Nabil, do you have any closing comments you'd like to add?
Yes, thank you very much, everybody. I'd like to, in particular, thank Dr. Lebo and Dr. Srecki for their valuable time and information that they provided us all with today. We are at the hour, so with that, I want to thank you all for joining us today, and I believe this session will be available to listen to or download from the website as well, the Becker's website. So thank you very much, everybody. Appreciate it.
Thank you for that, Nabil. And that is all the time we have for today. I want to thank our SunQuest and our presenters for the excellent presentation, and to our audience for participating today. Enjoy the rest of your day, and we look forward to having you join us for future webinars.